It's a tremendous pleasure to be able to introduce Tim Engelhardt. He graciously agreed to give us this presentation today. I'm really excited that he's willing to do so. I met Tim when he sailed on Expedition 329 with me and a group of 100 or so of our now closest friends. And he sailed to, in large part, look at the viral communities of the subsea floor sediment in the deadest part of the ocean. And that's something that no one had done before. He's obviously, as you'll see from his presentation, gone very well beyond that. And I want to make a pitch here, and Tim can correct me, but he's been recently finishing up his graduate program, and I want to say that any institution that could have the opportunity to basically snap him up should do so, because he's just doing marvelous work. And he's a pleasure to sail with.
Thank you.
So with that, I will hand over control to you, Tim. I won't introduce your topic because I think you'll do that yourself.
Yeah, thank you very much, Steve. I think I have to do it like this. Some presentation on the screen now. No. Okay. Can you see the presentation? I think so. Yeah, the topic for today is viruses in marine subsurface sediments, and I would start with these few pictures here. So what you can see here is a cell in isolated from marine subsurface sediments, and what you can see here is phage attached to the cell surface, and here you see phage particles inside this cell, and here you see the free phage particles after the life of the cell. So, but let me start with a brief introduction. So, when we're talking about the marine deep biosphere, we are talking about the largest interconnected global habitat, and it comprises a variety of different environments with extreme conditions, sometimes with extreme temperatures, but also we have energy-rich and also very and extremely oligotrophic sediments. The life or a summary of the cells of the life in the marine biosphere was given by Karlmeyer and colleagues recently. And what you can see here is the number of cells in the sediments. So the cell numbers is basically decreasing with depth, but also decreasing towards more oligotrophic sediments. So this is basically mainly continental margin sites, and this is, for example, this South Pacific gyre here shown in yellow. Based on this, we have a global estimate, which is about 0.6 on average percent of the global biomass that we can find here in marine deep biosphere. Cells were found as very deep sediments, as deep as 1,600 meters below the seafloor, and most of these cells have to deal with low energy fluxes and with energy limitations, so they are basically energy starving. This is mainly due to the recalcitrance of organic matter in deeply buried sediments, and based on this, the doubling times or the turnover times of prokaryotic carbon is estimated to be between years to centuries. Interestingly, the cells are alive and they can deal with this energy limitation. And what I would like to talk about now is another factor in the marine deep biosphere, which is causing prokaryotic mortality, and probably also have a positive influence on microbial communities. So these are viruses, of course. So first of all, when I'm talking about viruses here, I'm talking about bacteriophages. These are viruses that infect archaea and bacteria. And there are no real life forms, and therefore they are described as biological entities, as the smallest biological entities, and the most abundant biological entities on our planet. So here, on the right side, you just see an illustration of a famous phage, the T4 phage. What I wanted to point out here is that the phage mainly consists of the viral genome, and the major part of the virus particle is made out of proteins. In total, we have about 10 to the power of 31 viruses on our planet. And what I find very interesting estimate is that we have in the oceans 10 to the power of 23 viral infections per second. If we talk about viral infections, we have two main viral life cycles. So the first is the lytic cycle. You can see it here. So the phage is infecting or absorbing on the cell surface, and the viral genome is introduced into the cell, and the metabolism of the cell is used to produce new phage particles inside the cell, and at the end the cell is lysed and new phage particles are released. On the other hand, we have a lysogenic cycle. In this case, the viral genome is integrated into the host genome, and the replication of the phage occurs during cell division. If you're talking about the sediments, we can ask the question what reproduction mechanism is more likely to occur. And if we think about highly compacted sediments and in most cases very low host cell densities, the lysogenic cycle is less likely than the lysogenic cycle just because of the low rates of host cell contacts. I will talk about lysogeny in more detail later on. First a bit about viral ecology. So the points why we are interested in phages is their huge impact on microbial communities. So there are mortality factors for bacteria and archaea, and depends on the importance as mortality factor depends on the environment we are in. So for deep sea, for example, they are contributing up to 71% to prokaryotic mortality. And if we go to deep sea sediments or the surface of deep sea sediments, they can contribute up to 90% to prokaryotic mortality. This is a top-down control of microbial communities. They shape and diversify these communities, and this is basically described by the so-called killing the winner theory. Another point is so-called viral shunt. This is how viruses influence the microbial food web. You see it here on the right side. The prokaryotes that are lysed, the cell deposit is released after cell lysis is filling up a pool of dissolved and particularly organic substances, and this is again usable or utilizable by the surviving prokaryotes. Here you see a nice picture. This is lucky shot, so to say. What you see here is a cell that is just lysed during one of my experiments. What you see here is stained DNA that is released from the cell. If we go to the sediments, this was shown that for the surface of deep sea sediments, the viral shunt is releasing or recycling 80% of the bacterial heterotrophic production. And another study by Middlebury and others showed that in the first centimeters of the sediment, up to 140% roughly is removed by viral lysis again. There's one more thing I want to point it out. This is viruses as agents of gene transfer agents, so to say. They mediated gene transfer, therefore drivers for evolution. And what you see here is a process of transduction on the left side. Here on the left side you see the lytic cycle again. So what can happen is that during this phage maturation process inside the cell, the host DNA is accidentally packed into the phage particles, and after release of the phage particles, this results in a so-called transducing agent. During the lysogenic cycle, it might happen that parts of the host cell chromosome is accidentally packed into a phage particle, and also resulting in a transducing agent. And by infecting new cells, the genetic material from the a former host cell might be integrated into the new cell and the genome of the new cell. By this process, phages might, or phage-mediated gene transfer might alter the phenotype of the cells by introducing a fitness factor into the cells, which could be resistances, for example, or membrane proteins for substrate uptake, things like that. Another interesting point is that viruses can also shut down genes and influencing the host cell metabolism, especially if we go to an environment where that is energy limited. The gene shutdown might help to save energy, which might be very important in marine biosphere and the sediments. So, conclusion, phages can increase the survivability of host cells. The thing I want to start with is the very first experiment I performed on phages. From the beginning, we thought that if we screen isolates for the occurrence of phages or prophages, is a good way to isolate the biosphere-derived phage host systems. So this is one of those induction experiments. In these experiments, you are calling prophages to be induced and to form free phage particles. And what you can see here is a growth curve. So the optical density is the cell density over time. And what I did is I grew this culture to the early exponential growth phase and split the culture to 3 parts at mitomycin C to 2 of these subcultures. Mitomycin C is a DNA damaging agent and is inducing prophages. What you can see here is that the 2 mitomycin treated cultures, the cell density dropped after about 6 hours. After the addition of mitomycin C, there is a control showed further growth. And now you can also see it here by naked eye that at the end of the experiment, the mitomycin treated culture is clear, in contrast to the control culture. Virus particles per milliliter were 10 to the power of 10 roughly in the induced cultures. What we did in the first study is he shows a variety of different isolates from different phyla and performed this induction experiment. And what we found was that 6 out of these 13 strains we tested harbored inducible prophages. And this is, here you can see the phages that we induced. And this was a great moment actually, because this showed the presence of functioning prophages in the deep biosphere isolates, and therefore the first functioning deep biosphere derived phages. We want to analyze this in more detail, the occurrence of lysogen, and we chose a population of a deep biosphere organism, which is Rhizobium radiobacter. And the reason for that is that we have this organism was the most frequently isolated organism we have in our culture collection from deep subsurface sediments. In this case, these organisms, in total 36 strains were isolated here. This is Leg 201, this is a Peru, continental Martian, equatorial Pacific. And the first thing we did is we quantify these organisms directly in the sediments by qPCR, by specific qPCR, and we found out that this organism contributed up to 5% to the total microbial community. On average, this was 0.75%. Interestingly, a similar abundance of this organism was also found in the Mediterranean. Thus we thought, or it is a good model organism just because of this of the abundance of this organism and having occurrence in different subsurface environments like Peru Martian, equatorial Pacific, and also the Mediterranean. What we did is we analyzed first the host organisms. What you can see here is cluster analysis of the ERIC pattern. This is a fingerprinting method to fingerprint the genome organization, to analyze the diversity below the species level, actually. And this is the first interesting thing we found out, is that we have in this population distinct subpopulations at the different sites. As you can see here, we have a subcluster for site 1225, this is Equatorial Pacific, and this two subclusters here are formed at site the Peru margin area. We performed induction experiments with all these strains, and it turned out that all of the strains harbored at least one prophage, so all the strains are lysogens. And here you can see the genome sizes of the phages as analyzed by a PFGE. And yeah, what you can see is that some of the strains do have a multiple prophage infection. And we work further on this, on the phages and phage genomes. This is what you can see here. This is the two-phage genome induced from one of these rhizobium strains. I thought that I'd get first hints here for some genes that might help the host organisms, but unfortunately 70% of the genes just have an unknown function. At least we are according to the annotation. So, but what you can see here is that you have certain core modules or core regions. So here, for example, all the genes encoded for the structure of the phage, and we have here we have genes that are involved in the DNA processing, and we have also some genes that for the regulation of the phage. I want to point out one gene. This is this one, the anti-repressor gene. This gene is involved in the switch of the viral life cycle, so the switch between the lytic and the lysogenic cycle, and thus it's specific for temperate phages. And I used this gene to create a specific detection system of this gene via qPCR. And what I did is I quantified this gene in the original sediments. What I found out is that we have between 10 to the 5th and 10 to the 6th gene copies per cubic centimeter of the original sediments. And if you compare that to the number of phages here shown in red, it turned out that up to roughly 14% of the phages that we can find in the sediments do harbor the anti-repressor gene and are therefore potentially able to enter the lysogenic cycle. So what we have at the end is a very abundant and widespread population of Rhizobium radiobacter, which is completely infected by prophages. And we also found up to 40%, which is a huge amount of prophages in the free phage fraction in the sediments that do harbor a prophage anti-repressor gene similar to that found in the rhizobial phages, which basically indicated that lysogeny plays an important role here in this community and also in the sediments. This is actually the first time I talked about the viral abundance in the sediments, and this is where I would like to go on. So during the last year I analyzed a lot of different sediments for the viral abundance. Here on the map you see the different locations I analyzed. So this is again Peru margin and Equatorial Pacific, so leg 201. Basically what you see here in red, this is the German tidal flat. This is sediments of very high microbial activity due to the very high input of fresh organic material. Here, this indicates the Bering Sea. This is site 1344. It's located in the area of the green belt. So we have a high primary production in the water column above the sediment, and high sedimentation rate, resulting in relatively young sediments. And here, shown in purple, we have the South Pacific Gyre. This is the opposite. So we have a very low primary production in the water column and a very low sedimentation rate, resulting in extremely oligotrophic, probably the most oligotrophic sediments and very old sediments. If you're searching for viruses in sediments, electron microscopy is always a good tool to see if there are viruses and if the particles you're looking for are really viruses. Here you see some examples of some pictures of the viruses are extracted from these sediments. Here you see in this line examples of the tidal flat sediments. But interesting are the phage particles here in the middle. So this is the site in the very center of the South Pacific gyre. So we have the surface on the left side, about 10 meters below seafloor, and the picture in the middle, and the picture on the right side represents a phage particle right above the basement. So this is referring to roughly 40 million years old sediments. If you want to quantify these phages, you use another tool. This is epifluorescence microscopy. So here on the right side you can see an example. This is viral extract. Counting of viruses is quite similar to counting of cells. So this is an example of the Bering Sea, 33 meters below seafloor. So, and here on the left side you can see the abundance of viruses in these sediments I just talked about or showed you on the map. So what you can see is that the abundance of viruses with sediment depth is basically or generally decreasing, but also among the different sampling sites or areas, the abundance of viruses varied between 4 to 5 orders of magnitude, but it generally follows the trend of the cells. If you compare the cells and the viruses in these sediments, we'll end up with a quite good correlation. So as you can see here, the color code is still the same. So in red is the tidal flat area, and this is the equatorial Pacific, and the Bering Sea, the Peru margin shown in green, and in purple it's the South Pacific gyre. What you can see here is that every microbial community is able to support to support a viral community of a certain size. Also environmental factors play a role. We're talking about the viral community size. It can be seen here. And the color code is still the same. So in red shown the ratio between viruses and cells for the tidal flat sediments. What you can see is that we have a rather constant ratio of about 10 for this sediments. If you go to the equatorial Pacific area, Peru margin, and the Bering Sea, for example, here shown in green, we see that we have an increase of the virus to cell ratio in the deeper sediment layers from 1 to 10 at the upper sediment layers, and this is increasing to up to 20 for the deeper sediment layers. What you can also see here, shown in purple, is the virus to cell ratio from the South Pacific gyre. You can see that we have a dramatic increase with sediment depths, reaching a virus to cell ratio of up to 220 roughly. I think, the constant ratio that we can find in the tidal flat area, I think is similar to what we know from the water column, for example. So we have a very high productive area, and we have high viral production in the range of hours to days, and regarding the viral turnover, and we also have a high viral decay. So we have kind of a short-term balance between viral production and decay, constituting a constant ratio of about 10 between viruses and cells. For the deeper sediments, we see two things. The first is that we have apparently better preservation of viruses in deeper sediment layers. This might be due to the absence of X enzymes, for example, as an important viral decay factor in these sediments. But I also think that increasing virus to cell ratio with sediment depths is indicating that there is still viral production going on. And this can be seen here. So what you can see here is the number of viruses plotted against the age of the sediment. Here an example for the South Pacific gyre. What I did is, I'm sorry, so this is a power law function, and what I did is use the first order derivative to calculate based on this the decay of viruses at a certain sediment age, and you can see it here in the table. The first thing that we see is that we have a decrease in the number of viruses or the decay rate and the relative half-life of the sediment at 1 million years. Between 0.5 and roughly 2 million years, and this is increasing with increasing sediment age, showing that we have a better preservation of viruses with sediment depths. On the other hand, if we see, or if we have a look at the viral half-life, or community half-lives, at basement age or at older sediments, I think the time is quite unrealistic long. Of course, we need to keep in mind that in this calculation we excluded viral production completely. So what I think is that we have indeed better preservation of viruses with depth, but we also have a viral production that overlays the viral decay in the deeper sediment layers. Yeah, there's one more question I would like to ask here, and I know that we need to be careful with that. So what you see here on the left side is again the ratio between viruses and cells. And the question is if viruses can be a source of organic carbon by itself. So if you take the most recent numbers for organic carbon content per phage particle, which is 0.2 femtogram per phage particle, and for cells it's 14 femtogram per cell. The biomass that is viral bound actually would exceed the prokaryotic biomass with a virus-to-cell ratio of 70. So and this is happening here, especially in the extreme oligotrophic sediments of the South Pacific gyre. Based on this calculation, you would end up with a contribution of viral biomass up to between 50 and 80% to the total biomass. And in other sediments, with a lower ratio, viruses still contributed up to 30% to the total biomass. As I said, we need to be careful with that because in a slight shift in the cell numbers and the viral numbers, and also the amount of organic carbon per cell and phage particle, this calculation would change, of course. And okay, let me sum this up. So the first thing what we saw is that we have a very abundant community which is infected by prophages, or all this available isolates from this population are affected by prophages. And we have a good indication by quantifying a gene that is involved in the switch of between lysogeny and the lytic cycle, that lysogeny is widespread and quite common mechanism for this population, and also seems to be to make up a part of the free phage particles in the sediments, showing the importance of lysogeny. The second thing is that we have high numbers of viruses in the sediments, and the viral numbers always exceeded the corresponding cell count. Subsurface sediments showing in general the important or the potential impact on the microbial communities just due to the sheer numbers. And the last thing is that viruses potentially contribute to the organic carbon budget of the marine biosphere, either by lysing cells and by providing organic carbon after cell lysis, or even by providing organic carbon itself. So, and with this, I would like to thank you for your attention, my colleagues and my supervisors, my co-authors, and also the funding agencies, my group.
All right, thank you very much. So now, for anyone who has questions, please start typing them in the chat box, and Tim can answer them. Sure. All right, we have a question from Bill Brazelton. Would most methods count viral biomass as sediment VOC?
I think it depends on the method. I think viruses are defined as DOC due to their small size, and they are basically staying.
Rika has a question here. Gene transfer agents, GTAs, are phage-like entities that have been seen in various archaea and bacteria, particularly in the alpha proteobacteria. Since you were looking at phage from rhizobials, I'm wondering if you looked at these phage genomes for genes similar to what has been seen in GTAs isolated from other alphas. This seems like it might be a really interesting environment in which to find GTAs, considering the abundance of lysogenic phage in the deep biosphere.
All right. I actually didn't look in particular for genes, compare that with GTAs. So, yeah, you're right. I think it is interesting to compare the viral genes we have, or the genomes we have, with other genome data. And yeah, I'm actually not sure if these organisms or the Rhizobium radiobacter do have GTAs. I think one genome is...
okay, Myrna Jacobson-Myers asks, can you directly measure decayed virus as well as...
It's a tremendous pleasure to be able to introduce
Well, I think to measure or count intact viruses in the sample, I think this is what we are doing by counting viruses. I think you see the small dots and you can count them. To measure viral decay, I think it's possible to you can kill the cells. There are some methods. The problem is that, especially for sediments, what you do is you dilute your sediments in preparing a slurry. And as far as I know, this might cause an accelerated viral decay just to by disturbing the sediment or
I see Myrna's typing. Any other questions?
So, but it is possible, and I think people are doing this. Yeah.
Myrna says, thinking of a mathematical model for production, decay, and lability, etc. From the Dot Lab, can you estimate the half-life of a viral particle?
Yeah. Yeah, this is an interesting question. So I don't know if you're talking about the maximum life of a virus particle. So I think the actual half-life of a virus particle really depends on the decay factors the particle is exposed to. So I know that viruses, I don't know how stable viruses can be in the environment. So if the environment is quite stable and the virus is not exposed to viral decay factors, it's probably absorbed to the sediment matrix, I think it might be stabilized in the sediments. I think it's difficult to estimate this, at least if you want to refer back to the environment.
Myrna asks, do you know what the major cause of viral lysis is in an environment?
Major cause of viral lysis? No, what exactly? What do you mean exactly? I think the viral production or the viral productivity, I think the most important thing is, depending on the environment, if you have mainly phytic reproduction, I think this is in the water column, for example, in the photic zone, for example, during algae blooms and stuff like that. I think the rate of phage host contact, this is responsible for the productivity of the viruses. If this is what you're referring to, and on the other hand, if you have a more lysogenic cycle or production via the induction of phages.
We have another question here. Have you looked for any lytic phages in this environment?
The frequency of induction events in the environment. I don't know if this was the question. Yes, I have actually. What I did is the strains that do not harbor prophages from my very first study. It was 7 strains. I extracted viruses from the sediments and trying to do or perform plug assays with these strains and the phages in rodent pore waters, but without success, unfortunately.
All right. Well, if there aren't any other questions, thank you, Tim, for the great presentation. We have Karen Lloyd coming up. Well, here's one perhaps coming up, but Karen Lloyd will be the next network speaker coming up July 25th.